Welcome everyone to the Friends of Curry Campus Speaker Series. This is the fourth and last talk that Bill's going to give on the marine invertebrates. I hope you guys that have been coming have enjoyed it. Hopefully there'll be more planned in the future. We'll see. Let's see. Let me go over first our lineup for fall. Let's see, August, next month, we don't have anything. September is going to be the Audubon Society will be here again talking about the backyard birds, how to get backyard birds into your yard and what to do about predators such as cats and things like that. The following month would be September, Dennis will be talking about the monarch butterflies and the new technology on how to track them, which we'll be able to do on our phones as well. November, I'm going to be talking about the Australian rainforest, the flora and fauna, mostly some of the birds, the butterflies, and the plants, which are really unusual in the rainforest. And then in December, we're going to have a Christmas music program. I'm not sure exactly what we're going to have, but it'll be a couple of different things, I believe. So, okay, so that's that. I want to talk about the cookies because I always forget to mention them. If you've been here before, Melanie has done a great job making cookies that fit the theme of the talk, which I told her no one really is going to care, but she's been very specific. If it's starfish, it's got to be starfish. If it's crabs, it's got to be crab. So she is not here. Unfortunately, she's out of town, and so the cookie baking fell to me. And I am not the baker that Melanie is. So she left me a cookie cutter that is a jellyfish, which is one of the things we're going to talk about. And I decided that all those little tentacles would probably just break off. So I took the easy way and I made chocolate chip cookies instead. And I think you can all just pretend that they're jellyfish with no legs. But Bill said there is probably something out there in the ocean that looks like a chocolate chip cookie. So.
And I'm going to show you a video of it.
Yeah. There you go. So anyway, help yourself. Karen has also made some gluten-free cookies for those of you that can't have gluten. What else? Oh, I wanted to mention a couple of things. Karen mentioned last week about the marine biology program that they do have here at the college. If you're interested in donating to a scholarship, you can see Karen. She's there in the back. And she will be happy to help you with that. And I also wanted to just mention how thankful we are to Jill for putting us in this big room, and for John, and for Alita, and Michael, helping out so much, with all of this. And also Michael in the back, who's been recording. So these, if you can't see the talks, you can see them on YouTube. So I think that's everything. So Dr. Bill will start his talk.
Thank you, everybody, for especially those of you that have tolerated or you stuck around for all four of the talk. I've tried to make them as interesting as possible because it's trying to reflect my tremendous appreciation for the marine critters that are there. I think I'd mentioned that I was trained by the people that put together the Monterey Bay Aquarium. Everything was fresh stuff, which made it hard to get through the labs because you're just so fascinated in these critters. I came within this close of not being here today because I was in Washington, D.C. on Sunday, Monday, and Tuesday talking to our two coastal representatives and one of our state senators and our senator and one of the senators from Hawaii about protecting our oceans. And so with that, there was an interesting thing in terms of who is concerned about climate. The hat I wear is all about climate. So that's a big thing for me. if you could put a check over in their corner on that table that talks about the 6 different types of people in terms of their concern about climate, that would be good. One, just so I can have a sense of here at Brookings, how many people are concerned. Also, it's for you to see how many of your peers have interests in different places. It's usually a surprise. People said, oh, I'm not alone. There are more people that have these interests. So if you could do that, and you'll get a cookie if you do. Anyway, you know. The other is that we're going to have a talk for Coastal SoCan, Southern Oregon Climate Action Now, that will be by Ann Velisis, who is the president of the Kalmiopsis Audubon. She's going to be talking about proposed potential nickel mines here in the county. So if you're interested in that, take a look at the flyer over there. If you want to get it, because this is all be on Zoom, so you don't have to be here or at the library, you can give me your name and your email, and I will give you the link. I'll give you the way to get onto the thing. So those are the 2 announcements that don't have anything to do really with these things here. So why don't we go on, and we're going to be talking about the invertebrates in the sea. All right, we've seen that before, no surprise. And you've seen most of these things, how we're finding out that only 3.5% of the known animals are us, vertebrates. And so we're doing the others that are non-vertebrates, invertebrates, and yet even with that, over half of those are terrestrial, that are insects mainly, with some spiders and such, all that. If you look at, we had mollusks the first week, crustaceans the second week, the corals and such this week, with the echinoderms thrown in there, and that's really only 8%. And that means that there's another 38% that are other marine invertebrates, and that means all marine invertebrates are a little less than half. And then I've shown you this, that we only know 3 to 5% of the animals that are out there, the marine invertebrates. I had somebody, actually it was Melanie, who said, how do you know you only know 3.5%, 3 or 5%? If you noticed, a lot of those really good videos were from MBARI. That's the Monterey Bay Aquarium. They are using ROVs, remote operating vessels, to look around what's in Monterey Bay. And they keep finding new animals, new descriptions, new things, just in Monterey. And then they say, let's expand it out to the rest of the ocean. The ocean has the largest habitat anywhere. You think it's very, very deep. 70% of the Earth's surface is marine. And so the fact that we know little, we keep seeing new stuff, that's how they estimate we only know this much. Which, again, for me as an ecologist, I get concerned about saying, well, let's go do deep sea mining. Let's go shut down this, let's do that. I want to at least know we've got some science behind it, what we're going to make some smart decisions. So, with that PR, we're going to be talking about corals and jellies, worms, sea squirts. They're going to tell you which of all these things are most closely related evolutionarily to us, taxonomically to us. So we're going to say, what are the major groups of the 2 major phyla that we're talking about and some smaller phyla in terms of the numbers of species? Common characteristics of each, how they feed, how they reproduce, how they are surviving in extreme environments. So we're going to start off with the Cnidaria. The Cnidaria, that group is named because they all have a specific type of stinging cell. We're going to talk about that, how it's special. Now that includes the corals and anemones, the jellyfish, hydrozoans, which are really bizarre, but you actually do know about them, and cubozoans, which are really mean. Going to also talk about the tunicates. They're salps and larvaceans. Those are the ones most closely related to us. Talk about worms, the annelids. You're familiar with the many worms here. There's nothing that matches what's down in the ocean. And those are called the polychaetes. Polychaete, many legs. Talk about the ctenophores. Those you're probably not likely to have ever seen. They're comb jellies, and they are absolutely incredible. They're so beautiful.
And then we're going to have some bryozoans. I'm going to have to convince you that they're animals and not plants. All right, the cnidaria, that includes the anthozoans. Anthozoans are the flower animals. So we have anemones, that's pretty clear. Hard corals, if this is a day shot, if it was a nighttime shot, their tentacles would be out. So night diving is really interesting in the Caribbean because it's a whole different world. A lot of things come out then.
Is that a brain coral?
That is a brain coral, very good, yes. And so that's a hard coral. We also have soft corals. You might be aware of sea fans and such. Now the jellyfish are the scyphozoans. Scypha apparently is Greek for cup. Turn it upside down, there you got your jellyfish. Hydrozoans are weird because they're a combination of kind of the polyps of the anemones and kind of the jellyfish cup things that are the jellyfish. And so here's a Portuguese man of war. You see there's a lot of complexity in there. And then we get the cubomedusae. You probably have seen the masks that some of the, I believe, I've never been there, but to Mexican wrestlers wear these masks. That's kind of, it just looks mean. Believe me, they are. All right, what are the characteristics that you've got with these guys? Typically, we're going to see some tentacles. That's very common in the anemones and friends. They have a mouth that's in the middle of that ring of tentacles. We have a stomach that's in the middle of this column. They're going to have some stomachs down here. We have the pedal disc. That's basically its foot. That's how it holds down or crawls around. They do crawl, not fast. Now, what's interesting, and I was talking about this, the stinging cells. This is a cell, it's a cnidocyl, but more importantly, it's this little organelle inside, which is the stinging part. That's the nematocyst. Now, the nematocysts are really well designed, if you will, in that they are like this. So here is the cell that holds the nematocyst in here. And if you think of it, if I can get my prop to work, the way that this works is when the little trigger gets triggered, it makes basically the cell shrink together. So it squirts the thing out. But just like, if I can do this, if you have I borrowed this from TSA yesterday. If you squeeze this, you'll notice that it doesn't go shoot out like a spike. It actually comes out and unfolds, and it unfolds. And doing that, all of these little barbs mean that it pulls it open, so the next part can go farther in. So it'll go farther and farther in. And by doing that, you get it popping out, and it go way deeper. And at the end, that's where they injects the venom. A lot of them, you don't really have much there because it's not that strong. Others, those cubomedusae, have very, very potent nematocysts. All right, they occur on the tentacles. You'd expect that. They occur in these mesenterial filaments, which is in the middle of the stomach, which if you're going to eat something, you'd want to have some way of calming it down, making it not really cause trouble. And then they can occur down here. There's little filaments, a concha. So what the anemone can do, it can squeeze itself together a little bit, and it's got little weak spots in its walls. And those concha go shooting out. So it has a way of protecting against predators or something that wants to come and nibble on its side. We have also zooxanthellae. Zooxanthellae, we'll see especially, probably familiar with that with corals. And they are little pieces of algae that give the green anemones the color of green. Now, we'll see in a minute how you can have a couple of different kinds of anemones next to each other and they go to war.
Each animal prepares for battle, inflating a series of sacs,
Special
fluids for combat.
nematocyst-filled sacs.
Inside each sac are hundreds of thousands of weapons. It is a veritable silo of warheads loaded with toxins. But in order to deploy its weapons, each combatant must reach out and strike its enemy. With each strike, thousands of poisonous weapons unleashed, penetrating flesh, pumping soft bodies full of toxins.
So you can see that these are well adapted, well ready to defend itself and to defend anything that comes around by it. Now, in reproduction, you have sort of the routine things. You have males and females, and they can release sperm and eggs. Typically, it's broadcast fertilization, which means that you better put a lot out there because you're missing a lot. It's not internal. You go through the embryo, the larva, you get a juvenile, and it grows up to a big one. Pretty straightforward. Then again, they do, different ones will do asexual reproduction. This particular kind, the frilled anemone, basically push off part of its pedal disc, and that grows up to a new individual, genetically identical to the original. The glass anemone says, well, instead of just a part of the pedal disc, let's make a whole little animal next to me. Again, it's genetically identical. And then again, it's always easy to just split apart. And then you have a whole series of them that are identical. Now, we just saw that video of one anemone battling another. But if you're identical genetically to your neighbor, probably not going to fight, not like kids. But if you started out and you have an anemone here and it reproduces a whole bunch and everybody is identical there, and a different one is next to it, when they come together, they're going to battle. And if you happen to see one, and we'll see an example of it, they will have a no anemones land in between. And they will, you can watch them as they battle for their place. So here's a real common giant green anemone that you find here in the tide pools. You probably have all seen those. And those are individuals. That's an Anthopleura xanthogrammica. Its close relative, Anthopleura elegantissima, and you can see why it's way more elegant than the xanthogrammica. But these guys reproduce by binary fission. So probably all of these are genetically identical. And yet if you see two of these that are coming from different parents, that's where you can see the little battles and see little lines between them. Other kinds of anemones, here's a corkscrew anemone, and it's apparently got a fairly mild sting for humans, but it's really nasty to other anemones, and it's really susceptible, or it's a lot of other animals, the crustaceans will be affected. But here are four examples of little shrimp, cleaner shrimp, that they get protection from the anemone, and the anemone gets something out of it, I guess. But it's interesting because remember those cleaner shrimp that will help take those parasites off the fish, which really helps the fish a lot? They are able to be in that area. So there's probably something from the corkscrew's benefit. Here's another anemone, a tube anemone. It tends to live in sandy or muddy areas. It's not actually a real anemone, depending on what the taxonomists say, because unlike everything else, it has an anus, and it has it at the bottom. Another interesting story is there's a particular nudibranch that really likes this thing. And so there are pictures of them, nudibranch coming up, starts nibbling. The anemone doesn't really like it, so pulls down, pulls the nudibranch in. Nudibranch keeps eating. When it's done, it comes on out, and the anemone just regenerates those tentacles. So a benefit of regeneration. Here's something, if you have a chance to go snorkeling or scuba diving, especially in central Southern California, you get these strawberry anemones. They look very, very much like corals, except they don't have a coral base. And so they're halfway between an anemone and a coral. And there's this one, the pom-pom anemone, which is really weird. It doesn't have a base. It's called the tumbleweed anemone, found in deep water. and it just floats around. It's kind of interesting. Here's a little coral, anemone down by it. But those guys, if you remember was it the first or second talk about the sea spiders? They love these things, and they'll take their chelicera and go in and suck out the juices from these guys. So the pom-pom anemone is odd. Many of these are. Okay, that's the anemones. Now the hard corals, because they have the good calcium carbonate structure, which makes up the Great Barrier Reef and other large structures. Staghorn coral, that kind of looks like a staghorn. We got the brain coral, that's pretty easy to see. The mountainous star coral, the big thing about this is you can see it spreads out to very large sheets. So it can occupy a large area. You can see the individual little spots there. You can see in this one, the starlet coral, even easier. Each one of those is like an individual polyp. So that's why it's got real close relationships with the anemones. Now the elkhorn coral, it's a different species in the Acropora family. That's palmata. You can see it's by palm-like. Now the interesting thing, both of these fairly fragile, and so they can break off. But the nice thing about it is that even they break off, they can reattach and they can grow. I spent 3 months in Hawaii trying to reattach corals when the U.S. Navy sent one of their, I guess it was not a destroyer, what's the smaller one that, one of the ships that's supposed to shoot down missiles. They missed the Honolulu harbor by 2 miles. And so they ran it up on the reef that was right in front of the Honolulu airport. That wasn't a good show for the Navy because it happened just a few days before the Pro Bowl. And so everybody was looking at it. They started pulling it off sideways and decided that wasn't working. Pull it off where it went in, that was good. Anyway, I was there for 3 months and we reattached 5,000 corals. And what was amazing is that within 2 weeks of putting it down and putting it out, detaching them was taking some concrete in a bucket, pushing it down, putting the pieces that were broken off because we had to collect all those. Within a few weeks, we had to go back to make sure we were looking at the right ones because we couldn't tell. So Mother Nature is very effective at keeping things going. Now look at how this is very similar to the anemones, because just like them, we have the tentacles at the top. We have the mouth between the tentacles, or in the row of tentacles. We have sort of the same thing, the stomach in the middle and everything. We have the column that's there, and we have kind of a basal plate, actually not a pedal disc. It is where the calcium carbonate structure really is. So this lives on top of that, which will make more sense in a bit than we see in the nematocysts. So they will have those for protection and other things. The life cycle, again, males and females, they'll reproduce in that way. If they break off, they basically are reproducing asexually. Here we have a brain coral. They keep coming up. You can see here where it's lost the living tissue. It's actually also over here. You can see it's a little bit whiter. What has happened there is the zooxanthellae, the little internal algae, have been pushed out. The animal, the polyp, can still survive as long as it doesn't go too long, because those zooxanthellae give it a lot of food. If there's no food gone, they can take for a little while. If it's too long, then the polyp will die. And once the polyp dies, it's not able to get rid of the things that land on it, algae and bacteria. And once that happens, the reef's gone, at least that particular animal. So it's going to be tough for that to come back for a while. So if you're familiar with the Great Barrier Reef, it's lost half its coral in the last 30 years, 30% since 2016, 10 years. It's in its 5th major bleaching in the last 8 years. So it's kind of like if somebody keeps beating you on the head, after you get over the headache for a while, but if it happens enough, it's not good. This is from a couple weeks ago from NOAA website. This shows, and you can see as it gets farther from the blue and more into the purple, that happens to be the equator. Also, you've probably heard about this El Nino, extreme El Nino. That's measured from here. And you can see just from the standpoint of the animals, the corals, that's where you're having the maximum chance of bleaching happening. I traded a week in the hospital in April for what would have been a week down diving in the Caribbean, but it was going to be, to get the orientation right here, and I was concerned, especially as a marine biologist, that's a fairly high level of concern about bleaching. So here's a report from 2025 about tipping points. Again, I wear that climate hat. If you look at that, there are a whole series of things that are, if you push it too far, you're going to fall off. It's sort of an odd way of comparing it, but if you were sitting in the bathroom tapping on the hand of the toilet, you can tap it a little harder, you can tap it a little harder, and once you tip it enough, everything's gone. I'll let you figure out what all's gone. But of all these things, here's like Amazon rainforest, it has to get that high. The collapse of the AMOC, the circulation around the Atlantic. Over here is the die-off for coral reefs. Its top part is at 1.5 centigrades change. We are at 1.5 centigrade change. I've got children, grandchildren. I will never be able to show my grandkids what it looked like when I was young. And so that becomes very personal for me. So just why I get passionate about this stuff. And it's not just temperature. Here's a crown of thorns starfish. We talked about that last week. Fourth recorded outbreak. They eat coral. A big problem in the Great Barrier Reef.
So catch snails in your garden.
Yeah. And that's on top of the bleaching. So it's a double whammy. So again, I've dived at the Great Barrier Reef and it's beautiful and I'd love to have it stay that way. Okay, moving on to the soft corals. They are very similar. If you could look up close, they have their little polyps, only instead of having a hard calcium carbonate base, they have like a proteinaceous base, so it can be flexible. And you can see these are named sea fans for a good reason. Some of the sea fans are sort of broader, and then, so you can get a lot of morphologies. Sometimes they look like fingers. How they got dead man's fingers, I'm not sure, but you can see the individual polyps that are there. So again, goes back to the anemone, that polyp phase. And this is one that looks more leathery, but again, it's got the same texture or same types of, you could see individual polyps in there. And some of it is more broader. And if you remember from last week, we talked about this particular starfish that is very well adapted to eat these kinds of sea whips and other soft corals. And it actually has the pedicellariae, it has the tube feet that allow it to hold on and just sit there and eat until it's all gone. And so it can be there apparently months. I guess there's not that much to do except eat. But the benefit is that it allows for a new kind of hard substrate that'll then be used by other animals. So it's like, well, there are some positives in any of this. Now, the other thing that is really cool, you don't see them very often because it's a little more often deeper water, but the sea pen is in that same group, and they have a bulb down here and they push it into the sand or the mud. And then you can see that there are individual polyps on each of those branches, and here you can see kind of the same thing. So we get that theme of the polyps. All right, shifting over to the jellyfish. Those are called scyphozoans. I mentioned that's because short or Greek for cup, I guess. So basically you take the anemone and turn it upside down. We have tentacles, we have a mouth in the middle, we have on the outside, things are called oral arms. They may or may not be on any individual jellyfish, but we'll see in some cases they're massive. We have the bell, or kind of equivalent to the column, and we have the stomach that's going to be in the middle, and there's a variety of other things that are there. We have the nematocysts, and again, some of those are pretty powerful, some of them not. Adult, the life cycle, again, fertilized eggs, you have a larva, and then you get to a polyp phase. In other words, it looks kind of ish like an anemone, but its whole purpose is to make these little baby the jellyfish. And so you can see they kind of look like that. And here's a larva, and we'll go through around. Here's the planula. Here's this polyp type phase, and now it's budding it off. And that's what you get as a larva that then goes on to be the adult. And you can see it's got very nice radial symmetry. It's got either these or the gonads or the digestive glands. They have little radial canals to get food out to the end. You can see some bright spots, which are kind of rudimentary eyes. And so they can see where they're going, sort of depending on the animal. And so they do actually have a nerve net. So some of them will be able to kind of tell where they're going. Monterey Bay Aquarium was the first aquarium that figured out how do you take something that's always in the ocean and never bumping into something and put it in a tank that it bumps into the walls. They figured it out to be able to give it, it's common now to be able to see in aquaria that these are, the jellyfish are moving around. Now here's one that's decided that working around and trying to get your food is kind of tough. It's in the Caribbean. I worked on this guy when I lived in St. Thomas, and it sits in the mangroves. It just lays there, and it lets the zooxanthellae provide its food. It has some very potent stingers, though. And as you're putting one of these things in a bag to try to measure its photosynthesis and respiration, you feel like you're kind of bathed in nematocysts. Anyway, now, as we've talked about shrimp, talked about oysters, there are people that like to eat jellyfish. So if you want, I can get you the recipe. I'm not sure I'll have any takers. I won't need to sample what you make. So I picked this up on the beach over the last many years of walking the beach. You can see the zooxanthellae. This is a part of a jellyfish, the middle part. Probably it's the Pacific Sea nettle, which gives you an idea of, I was holding this, it was the top part of the bell. And so it didn't have nematocysts that I was affected by, but there's all this, here's the oral arms coming out, and there are the tentacles coming out, and it's called a sea nettle for a good reason. There are animals, leatherback turtles in particular, that love these things. In fact, they come up here to eat them. And that is a critical habitat for leatherback turtles. And so that's good, except that looks an awful lot like that, which are plastic bags. And sea turtles can't seem to tell the difference until it's too late. And the jellyfish does go through their system, the plastic bags don't. Okay, other kinds of jellies are, I'll pick some ones that don't look like the cookies. The helmet jelly is also known as the alarm jelly because when it's disturbed, it will bioluminescent and it will try to scare off the predators. In addition, it's got this kind of red camouflage. It is down in the very deep ocean, so there's no light there. So in the absence of light, red looks black. Well, the benefit of this too is that it's a predator. So it eats something, and if when it eats it, its food starts to bioluminesce, then this will keep it from being visible from other predators. And it's got this weird thing. It's a very rigid crown, and it's got the long tentacles that sit up. So it changes what you'd think of for a jelly. And it lacks eyes and a brain. How you don't know it, how you can judge that it doesn't have a brain is interesting. It's more morphology. And it loves small fish and crustaceans. Now here's an interesting one. The psychedelic jelly. Yep, it's not very big. So 2.5 miles down. So it turns out that jellyfish moms are good moms. So catching the videos are very difficult, and you can see how quickly they can get away. Here is your jellyfish that looks like the cookies.
Wrong color.
That's true. So it has different colors. The interesting thing is that it's huge, and it doesn't look always the same way. It's very mobile. Now I'm going to show you something that's going to prove that there are more than just me that are marine biology, marine invertebrate nerds. Like I said. Kind of like Casper.
Yes. It's got to be a choppy.
How big is that?
So you can with the lasers here, and then I'll turn them off.
So they're big.
Oh, it's so spooky. Oh gosh.
Every day is the 4, right?
Now they're deciding how exciting is. 4 out of 5.
Sorry, but it's got some weird hitters.
Oh, it had the geologic mesh.
Oh.
Which is characteristic of that particular species.
Oh.
I'm not sure where the crinode was. I think it may be up there, but that's the isopod. Imagine spending your life inside a big, huge jelly balloon.
Because they have a resident isopod, I believe.
Yeah.
So what's amazing, it's hard to see any of that. So it's a little bit clouded, but
This might be a 5.
see the lines that are coming down? Characteristic of some of the ones that we had seen before.
At least speak.
Just that's an isopod that lives inside. It's a passenger. As they said, it kind of looks like it's driving the jellyfish. So you can, if you remember the isopods, that kind of looks flattened and it's got the legs there.
Hello.
And the comment about
You,
its feet would pop through because it's so gentle, but it's careful not to do that. Anyway, so that's why I tried to convey to Vicki and to Melanie that you can do whatever you want with the cookies, and we're going to find something that is like that. Okay, now to the nasty ones, the sea wasps, well named, the cubomedusae. And these are interesting, the box jellyfish, another one. We lived in Australia for a couple of years, got to know some people there that were in the Darwin area, which is the northern part, which is nearer to the equator. There are big animals up there, crocodiles that are, from here to there, something like that. You don't want to mess with them. So the people in Darwin said, we love the ocean, we don't want to have to not go there. So they built a little pond. And crocodiles won't come into that. So they pumped the water in. The fatal flaw to that was that meant that they took these things and ground them up, threw them in the water, and they basically made fire water. Not in liquor, it was a matter of nematocysts everywhere. They didn't use that pond. And this is the one that I think just looks so nasty. Sting can be fatal. There was a show, Jackass, where people do stupid things. Somebody put this on himself and he almost died. So these are very, very potent. You don't mess with something that looks like that. Okay, hydrozoans. That's the next group, but it's kind of a mix of what we've already been talking about. You have the jellyfish stage, the medusa stage, and you have the polyp stage. And they get to be very complicated. And it's sort of, well, what is an animal? Well, that's an animal. But when you have a bunch of these things, which part is of the animal? It's actually all an animal. Very complicated one. You might have seen on the beaches occasionally this velella, the by-the-wind sailors. Sometimes they wash up in huge numbers. Oh, I was jumping ahead. The hydrozoans included the by-the-wind sailors, the narcomedusae, which are similar to the other ones in structure, certainly. We have the Portuguese man-of-war, and that's a siphonophore. You can see a very big difference between this siphonophore and that siphonophore. We'll talk more about each of those. Then there are hydroids, and that's the one that I'm going to try to convince you that's an animal, not a plant. And then the hydrocorals, which they have some nice ones and some nasty ones. So those are not really a true coral, but they are this mix of lots of different types of individuals within that group. So now on to the velella. I'd seen somewhere on Facebook where people were complaining, who threw out all that blue plastic stuff? It's like, no, it's actually an animal. And if you wait for a few days, it'll lose its blue color and it looks like it's kind of snowed. That's a quarter. And you can see they're in sort of that size. And that's an animal in my hand. So it does have nematocysts. It's bad if you're a tiny little crustacean, but it didn't affect me because it's not hard enough to get through our hands. A narcomedusae.
Meet the dinner plate jelly, Solmissus. This denizen of the deep has an appetite for other gelatinous animals. Jellies, cold jellies, siphonophores, salps, they're all on the menu for Solmissus.
We'll talk about most of those soon.
This jelly has one of the most diverse diets of all midwater animals. So far, we've seen Solmissus eating 21 different types of gelatinous prey.
So again, we have a variety of types. You think, well, that's pretty straightforward. Doesn't seem like more of a jellyfish than a hydrozoan, but it's got other characteristics that keep it in that group. Now, the siphonophores are some really interesting group. They are an interesting group, including the Portuguese man-of-war. And you probably, if you've experienced, I hope you haven't, but you probably have heard about, they have very nasty stinging cells. And even if it's washed up on the beach, it's still very potent. If you get it on you, you're going to remember. And what's even worse is if you're diving, you're swimming around, the tentacles trail on a long ways back. And I've dived with people that come back with welts on their arms that they didn't even know where they got it, but they knew it was from a man-of-war. So these things have a big sail up here. A nematophore, that's a specialized part. It has these gastrozoids, well, the gastro being for digestion, the gonozoids for reproduction. You have the tentacles or tentacular zoids for the capturing of prey and everything. So it's all these specialized organs that are acting as an individual, kind of as an individual animal in this whole colony. That's when individual, what is it?
Siphonophores are gelatinous structures composed of specialized parts growing together in a chain. A small gaps-filled float provides buoyancy, swimming bells provide propulsion, and along a central stem, other parts sprout for feeding, defense, and reproduction.
Whoa. 16 feet long. Incredibly complex.
Siphonophores can break apart at the slightest touch, so they're difficult to study with nets and other traditional tools. For remotely operated vehicles, or ROVs, give us a close-up look at these seaside faunifores without damaging them. Our still pilots carefully maneuver the ROV to record stunning video of these complex creatures. Video observations are an essential part of data collection for our scientists. Viewing these delicate animals in their natural habitat helps us understand how they derive energy.
So they sort of between 600 and 1,800 meters, between 2,000 and 6,000 feet. It's tough to go down and dive to see those. There are some really interesting things in the upper 60 feet, though, and a way to do that is do blue water diving. And blue water diving is really pretty neat. You take a big float that you have has a boat buoy, put a 30-foot line on that, tie that to a bar that's about this big, put a 30-foot line on each end of those and hook that to somebody's weight belt. And then you can be sure you're not going deeper than 60 feet because very often when you're there, all you see is blue out there. So you can't see if you're looking that far or looking that far. So you end up looking up and close, and it makes it easy because, as I said, you can't get those siphonophores or any of the little delicate stuff. It dies in the net, just gets torn apart. The problem with that is I was doing this off Catalina, and you don't want to listen to that little thing from, what's the theme from Jaws? Because you're in the middle of the Catalina Channel and you're 60 feet, that's the bobber, you're the bait. So fortunately it didn't happen, but it's this kind of thing, you don't think about it. All right, ostrich plume hydroid. So I was gonna ask you, where is the plant here? And that's actually a piece of kelp, the holdfast. And this is the hydroid that lives on it. And it's like, ooh, okay, that's kind of hard to say that it's got much of an animal side to it. But if you were to look at this up close, you can see there are these little branches coming up. And by the way, those are specialized zooids for reproduction. Look a little bit closer and you can see, yep, every one of those is equivalent to a little polyp. And you can see the diagram, it's a little bit easier, but you can see the individual polyp. So that's what makes up this thing, only you can't see it except under a microscope. Now the corals, the hydrocorals, again, they have a different morphology from the true corals, even though they do have a calcium carbonate structure to them. There are enough differences so they are in this hydrocoral group. These are pretty, they're common enough, especially on the seamounts off California. These are fire coral, again, very well named because you bump into those and their nematocysts are really nasty. And it's a good idea to stay away, unfortunately, because they can have different configurations. You don't really notice them until you really notice them. All right, now we're done with that phylum. We're now going to be talking about the comb jellies, the ctenophores. The ctenophores are interesting because they have a bunch of different characteristics. One of which is they have no anus. So when they eat, if they have to poop, they open the bladder, open the cell wall or the body wall, and they poop, and then it comes back together again. So it's kind of opportunistic where you poop. So you've got a chance to see a little bit of the color there, but
What is that?
again, bio nerds.
Ctenophore. There we are. I was waiting to see. You could see all these translucent teens they called cobras. You can see them. They're kind of rainbow colored, silver going down.
Can't really see them yet, but now you can see them at the side. See all those colors? There's sets of cilia together as teens, and they move up and down, and as they change, the light is reflected off, and so it is mesmerizing to watch the colors go past. And here's a really good example, a bloody belly comb jelly. Kind of has a fancy ring to it, but you can see how. And if you do a night dive and you happen to see one of these guys, it is captivating. You can end up using your whole dive just watching this little guy. All right, enough for the ctenophores. The tunicates. Tunicates are interesting. There are 3 different groups: the sea squirts, the thalassians, and the larvaceans. Tunicates are pretty basic. They're named because they have a a leathery outer shell, a outer layer called a tunic, hence tunicate. They have a branchial basket, which is basically equivalent to their lungs, and water comes in through, kind of like with a sponge. Stuff gets filtered out, and it goes to their digestive system. Water comes in through the oral siphon, out the atrial siphon. Now, we can have the digestive system there. Here's a lot more detail, so there is a lot more than anatomy to that. But the key thing is the larvae start out like this, and there's a notochord, and there is a nerve cord, equivalent to our vertebral column. So they have the characteristics of not quite a vertebrate, it's a hemichordate, but it's still a lot closer to anything else we've talked about so far. So there's the notochord, there's the nerve cord. One group is these guys. They are really pretty. That's a collection of them. Sometimes they're one individual, sometimes they're sort of stuck together. Other times it kind of is now starting to look like a sponge. And so telling the difference between a tunicate and a sponge sometimes is a lot harder than you might think. And here is one where it's kind of like that, only it takes off. And earlier they were talking about some of the animals that like to eat the salp chain, and that's it. And again, you can see if you were trying to sample this stuff with a net, dragging it through the water, getting that up to see what they are is a real challenge. Now, the pyrosomes, or sea pickles, are also tunicates. That's in the middle group that I was talking about. And there was an article 2 or 3 years ago where there were just thousands of pyrosomes that washed up on shore, and people wondered what they were. It's this basic thing. Again, it kind of looks like a pickle. It's a hollow pickle. These are individual zooids, individual animals, kind of like we saw at the very first slide, where it takes water in, filters it out through the branchial basket, and pushes out. Well, in this case, it pushes it out into that common area, the cloaca. And so that gives a little bit of a force out, getting rid of it. Probably a little propulsion forward, probably not a lot. But that's why people can look at it and figure it's weird. Interesting thing about it, this was from 2024, having to do with the marine heat waves that we're seeing. The suggestion is that it can cause a disruption of the ecosystem structure so that instead of favoring fish, it favors gelatinous animals, whether it's the jellyfish or the tunicates or otherwise, which is not a shift from our productive ecosystem out here that we would really like.
Meet the giant larvation.
And this is the weirdest of all.
The animal is actually the little tadpole-looking things. This is the body, and this is its tail. Bathycordeus is considered a giant among larvaceans. Take a look at this one. It's about the size of a breakfast muffin, but it towers beside the tiny, regular variety of larvation. I know they don't look like much. The giant larvation's claim to fame is the huge house it builds. I'm talking about that big mucus bubble that surrounds the larvation.
Remember all about marine snow? They're harvesting.
Here's one without a. In the different, the house is made up of 2 filters and basically functions as an elaborate eating apparatus. Eating is a safe process for these little guys. They eat tiny particles of dead or drifting plants and animals. They float through the water columns. Here's how they eat. The outer filter traps larger particles too big for the animal to eat, while the inner filter kites just the right size big particles into the larvacean's mouth. Look at it beat its tail, which pumps water through its house to keep those particles coming. Eventually, the filters get clogged, and the larvation Van den Sand. The sinking houses are packed with food particles. And that's a good thing. They transport much of the food to animals living even deeper.
You've seen that before.
If it weren't for the larvation to parting houses, animals like these
And that.
would have a lot less to snack on.
So it's interesting, again, mucus nets comes back and think that everything that's out there is probably going to get used by something. And it's a very interesting active cycle. Now let's jump over to worms. I know it's something that everybody's fascinated by, but trust me, when you start looking at the different morphologies and what they do, it's pretty interesting. Typical characteristics include many segments, and so it's repeated back again and again and again. They have a head, and oftentimes that head has appendages to it that are really interesting, some antennae, they have some eyes, they can often have, they tend to have the parapodia, which are like paddle-like things off each of those segments. And then they have setae, which in the setae are basically little spines, and sometimes they can be pretty significant. This picture just to show a kind of a diversity of what the different polychaetes can look like. Here's an artist's rendition of what they thought it could be, and kind of like with the cookies, probably if you think of some sort of structure that a polychaete could have, something does.
When you think of worms, you probably think of the creepy polylens you find on the sidewalk after a rain shower. However, in the deep sea, you can find an incredible diversity of worms living in an amazing assortment of ways. Many of these are polychaete worms that are cousins to the garden variety earthworm. Polychaetes have segmented bodies with paddle-like feet or parapodia on each segment. Most have bristles or chaetae that they use for defense, crawling, or swimming. Polychaete worms are common in the ocean. About 8,000 species have been described so far, but many new species are still being discovered. On the seafloor, polychaete worms burrow in deep sea mud, crawl along rocky canyon walls, colonize superheated hydrothermal vents, and even eat decaying well skeletons. But perhaps the most extraordinary polychaetes live in the midwater, swimming or drifting deep below the ocean surface, but far above the bottom. Floata and swimma are two remarkable worms that live in the
Creative names.
They swim by undulating their bodies while beating multiple paddle-like appendages. In the midwater, there is no place to hide, so these worms have protective shark spines as well as amazing glowing organs that may startle or distract potential predators. Believe it or not, this is another kind of polychaete worm. It typically drifts about 1,000 meters below the ocean's surface, collecting food particles using a mucus net that functions much like a spider's web. One of several new species recently described by scientists at the Monterey Bay Aquarium Research Institute, this worm was so weird that researchers had to use genetic testing to figure out how it fit into the tree of life. They named it Chaetopteris hugocorsinus. The species name means rump of a pig, because researchers
Kind of looks like the rump of a pig, I guess.
Another worm that collects food using a mucus net is Chiobius. Only about 1 inch long, this worm doesn't have any obvious segments, parapodia, or chaetae, yet it is still a polychaete. It wriggles vigorously when disturbed and is one of the most abundant animals researchers see in the midwaters of Monterey Bay. Dimopterus, the dancing bristle worm, is an active swimmer using large fleshy parapodia for bursts of rapid speed. These worms have transparent bodies to conceal them from unwary prey and are voracious predators on other drifting animals. They can also emit a brilliant yellow glow to distract potential predators. Dimopterid worms create round egg masses to protect their babies until the young worms grow large enough to break free and begin hunting in the open ocean. The next time you're digging around in your garden and come across a common earthworm, think about its deep sea relatives and take a moment to marvel at the remarkable diversity of polychaete worms. This will be Skiros learning at the Monterey Bay Aquarium Research Institute.
So polychaetes actually are really interesting if you can get into them. Shallow areas, I really like trying to take pictures of things, and the worms sometimes help. I like the fan worms in particular. These guys are interesting because they have all these radials here that have finger-like things and then cilia on those. They'll pull the water up from underneath, and as it goes past it, kind of like an airplane wing, it pulls it around so it gets caught. They will have the direction water flow is up. It has this pinnule section, a cross-section of that, and they have the cilia, so it's a very effective feeding mechanism to get the little stuff that's caught in the water into the food. And it's even got a mechanism to sort big food from little food so that it picks only what it wants to go into its mouth, which is there in the middle. Again, fan worms, I'm a big fan of fan worms. These guys are like a horseshoe. These guys are Christmas tree worms. And this is a treat when you can see something like this, except when you take a picture, that's what they do. Instantly are gone. So it's like, how close do you get to get that special picture and then get disappointed? These guys are interesting as feathered duster worms. They tend to live in the sand. This guy is nasty. It typically gets 3 or 4 feet. It's long as 10 feet. It's got as many as 600 segments to it, but that's not the worst of it. That's its mouth. It has these jaws, and those jaws have venom in them. And think of something that's 10 feet long with that sort of thing that can bite you. It's also known as a bobbit worm, and you might have heard years ago about somebody named Bobbit that had problems. So, it doesn't have eyes. It senses pressure. And it's got very potent toxins in that so that it can cut them up, and it instantly paralyzes them. So it is a very nasty worm. Fortunately, we don't run into that. What's that?
Where are those?
That I'm not sure. I've never dived any place where I know that they were around. I'm very glad. Makes that, yeah. And they mentioned about these guys that live in the bones of decaying whales. This is not really, doesn't do much. It does a little bit for getting air in, but below the surface there, it's got little kind of root-like things. Those have bacteria in there. Those secrete acid that dissolves the materials of the bones of the whale. And so then they have that bacteria that will metabolize that. And so this thing doesn't really have to eat. It just lets the bones decay, and it uses that. So again, everybody gets to use something.
Stay tuned.
This is about as extreme an environment as you can get. That's a 6-foot-long worm. That red is from hemoglobin. Same kind of hemoglobin that we have in our blood. So as the plate tectonics move, they disappear. And that's why you have to make sure you have a good reproductive strategy so that when your house goes away, your offspring can get to the new places. So, and that's probably more like by random chance than any directed effort. So these are the giant tube worms. They're really interesting. They have no mouth. They don't need the mouth because they extract the minerals from the black cockers, what this one's called. It's spewing out lots of very toxic stuff, including a sodium, no, I can remember sulfur. Thank you. And so we have the hydrogen sulfide that's being put out, and these guys use chemosynthesis. Plants use photosynthesis, and the plants use the energy from the light to break water apart and add it to carbon dioxide and make the sugars. These guys use the energy from hydrogen sulfide to do the exact same thing. So these sulfur oxidizing bacteria make the glucose, and so it doesn't have to do anything from there. And it's amazing because that's the fundamental basis for a massive, or large number of ecosystems in the deep ocean. Now here's an interesting guy. This is regarded as the animal that's got the largest thermal tolerance that's ever been determined. These are, the fuzzy stuff is sort of a fur that has a lot of bacteria on them. And those bacteria act as kind of an insulation, fuzzy, like a backpack. Fur coat, exactly, thank you. There we go. So those are the animals. Those are little worms. And that's scalding hot water. Yeah. These are very paper-thin tubes. They're only half a foot. But you can see how the water's shimmering, just how incredibly hot it is. So next time you think your house is too hot, especially here in Brookings where it really gets hot, could be worse. All right, let's see. All right, so I'm going to finish off with this one that shows a polychaete that kind of looks like a nudibranch. And so again, if you can dream it, Mother Nature's probably done it. The last one is another one that I'm going to try to convince you is an animal. You might have gone on the beach and picked up a piece of kelp and seen that little fuzzy stuff is on it. That's actually an animal. That's a bryozoan. And if you were to look closely at that, you kind of can see individual rectangles there. And if you could look at it when it's alive in the water, because it's not going to be alive, you won't see it up on land, but it's these little polyps that come out, individual ones. And so it's like, oh, okay, that whole sheet is a bunch of these. Now it can be flat like this, it can be frilly like that. There are lots of individual little polyps that are there. And it can be the other forms too. This looks like little pieces of popcorn that are there. But again, each one of those is an individual polyp and they kind of work together to share their resources. So we've talked about the cnidaria, especially with the stinging cells, the corals, anemones, jellyfish, hydrozoans, and those nasty cubozoans. The ctenophores, the beautiful comb jellies. The tunicates that are closest to us. the annelids, the polychaetes, which are really pretty cool, and the bryozoans, the moss animals. And so it shows a bit of the diversity that's there. And I think that that means I'm done. But I do remind you that if you want to join and hear Andalisis talk about what's being proposed for nickel mining here in the county, let me know. If you scan that, which is on the thing there, that'll lead you to a URL to join. If you forget that, you can just you can email me at bill@socan.eco. And if you forget that, there are probably enough other people around that remember better than I do those things. So, and that's where I'm done. And I'd see if you have any questions. I know I'm way overdue. My apologies. Yes?
Is that nickel mine say only via Zoom, or can you go to the library and you mentioned you see it?
Right now, it's only on Zoom. So we're going to try to push the SOCAN meetings because we're going to be talking about a variety of other things. I'm in negotiations to see about having the third, I think I mentioned it, the third episode of the Kiss the Ground movie here. If we can work out the logistics, that would be in person, hopefully here, but we'll see. Yeah?
I realize this is a 10-hour discussion, but just briefly, I was curious if you're for or against the offshore wind project and what your current understanding of the project is at this time. Has it been approved or is it on a hold? Do you happen to know?
I've been working on offshore wind since 2015, I think. And so I've been actively involved with this. I was on the committee for doing the roadmap and everything. I'm a scientist. I want data. I want to know what the project is. I want to know what the impacts are going to be. I want to have as much information as possible. I want to make sure that the proper information has been given out to people before saying I'm for it or I'm against it. Because right now we know this much about it and we have this many questions about it. So I'm not going to say one way or another. But I also, as a climate champion, and actually wrote a paper for BOEM on how to assess the benefits from offshore wind in an environmental impact statement. And it's not easy because there's no direct benefits. Like if you put a project out there, it's going to affect this, if I do something on land, it's going to kill out this many trees, it's going to have that. And out there, great, you put it out there, how many coal plants is it going to replace? No idea. What are the impacts going to be? So there's so much that's not known. I think we need to study it. Right now, there's nothing going on. If it comes back, it will take at least a decade before anything happens. So nothing is going to happen for quite a while. And certainly not when we have an administration that's trying to do everything they can to stop all alternative energy. But being informed and asking those questions is exactly what we need. So thank you.
Appreciate it.
Yeah.
Anybody else? Oh.
Is the nickel mining associated with chromium contamination at all?
That's a good question. I'm not sure how much. It all depends on what the resources are that they're tapping into, whether they have chromium as part of it. That I don't know. And that's, again, it's kind of like we're talking about the offshore wind. Find out what's there. Find out what they can do. And especially if you want to protect the environment, then it's the challenge. What are you going to do to make sure you don't have acid mine drainage? I worked on a project, if you've ever driven from LA to Las Vegas, at the top of the hill just before you drive into Nevada is the Mountain Pass mine. Spent 15 years working on that, looking at ecological and human health risk assessments. And it's not cut and dried. There's not a one answer. If it goes beyond 5, then it's bad. If it's below 5, it's good. So right now, again, it's because as a scientist, I want to hear what the questions are and I want to hear the answers. I've worked on projects where the proponent and the opponents work together to get a mutual solution that gets the objectives done without having big winners and big losers. So that's the big thing. But it's a really good question.
It's my understanding that Ann's program is going to be on the third at Gold Beach Library.
Correct.
And so you can go to the library and see it.
Yeah, thank you. That's true. Yeah, Ann and I sort of coordinated because mine was going to be Zoom. Then it's a different collection of people that can do it. I will say her presentation in person is way better than any presentation, certainly that I could give or other people on Zoom. So in person is always good. Thank you.
Hank Cunningham, I wanted to thank you for doing this very informative series. And I was wondering if you had an opportunity, you said you just returned from Washington, D.C. on a legislative trip, to mention this lecture series that you've spearheaded and how well attended it's been and how informative and how much we need to see more of this. And also to thank SWAC for being here for us to illuminate these issues. So thank you.
I firmly agree with all that. And yeah, I was actually mentioned that to Representative Bonamici. Because she actually called in by Zoom from Oregon to talk to people in Washington. Representative Hoyle was unavailable, but I talked with her representative and talked about that. Also talked with a woman that's in Senator Merkley's office and shared with her the poster that was the very beginning. And she was a marine biologist and was all excited to learn more about it. So it's word of mouth. And so to the extent that you want more of these, mention it to the people here. The people here are fantastic. Are they here today? So, no, it's very true. Yeah, there you go. So yeah, the logistics, if you were here at the first meeting, especially if you didn't hear the first meeting because you couldn't get in, having be so flexible to set this up and everything it takes a lot of work. And Alita and John and Mike really deserve a lot of credit for it. And my wife, who put up with hearing this multiple times, again and again, she kept taking the dogs outside because the dogs didn't want to hear it either. And the cookies. But because if you don't eat them, we're going to go home.
You've mentioned Monterey Bay Aquarium. Can you make some comment from a consumer's point of view about the Newport Aquarium?
Two times we've tried to get up to the Newport Aquarium, first off, it was COVID, and second off, it was closed. And so we haven't been there. And I'm very frustrated because I've worked with a number of people up there, and I'm excited to get there to then provide an in-person comment. From what I've seen, and it has to do with the work that I understand they had been done on the sea star wasting disease, which in that video, the vet at the Monterey Bay Aquarium, they got their the protocols for fixing or to treating the sea stars, the sunflower sea stars, from the aquarium in Newport. So I can't say from personal experience, but I really want to get there. So, yeah.
Just a simple presentation on August 4th, 5:15 to 5:30. What time zone was that?
Here. Yep. That's a very good question, especially with Zoom. Yeah. And that's why it's good to know. Yes.
Given our horrible track record of introducing new species places, is there a chance you could put wasting disease on the Australian reef?
The problem is it would not be focused specifically at the biggest problem there. Right now, it's actually bleaching. So that crown of thorns, if you make sure that it was specific to that, that might be an interesting aspect. But just as it wasn't just the sunflower sea star here that was affected, it was just one of about 10 or 20. And the one that was also affected was the ochre star, which when we walked the dogs this morning, we saw 2 or 3 dozen of those, something like that. And so it is too broad of a treatment for something that would have huge secondary effects. But it's a good thought, you know. Anybody else? Well, I want to thank you for giving me an opportunity to indulge my marine biology nerd self. And I so appreciated all the people that were going, oh, this is so neat. I go, yeah, it's cool. So thank you for that. That was fun. It was when we were thinking about it during the spring, thought, this would be fun. Maybe we have something to do in July, as if there's nothing else to do. So thank you. It was very much to meet you. Oh.